we'll just dig into our Osmometer Lab and demonstrations of diffusion. So we are now in lab. So I'm going to get the appropriate module up on the screen. And I'm going to share screen 2 here. Okay. And here we have Osmometer Lab. All right, so everybody submitted their PDF of the Osmometer Lab. And again, if we want to share our objectives, what is the objective for our lab today? Anybody?
Well, I have, what is the relationship between the rate of osmosis and the concentration gradient?
So that's exactly it. Those are our two main variables. We have on one side the independent variable concentration gradient across the membrane. And we're going to look at a couple of different versions of it. We'll have, distilled water outside and 20% in the bag. Then we'll have a different concentration gradient in the other groups, which is going to be a higher gradient, which is going to be between the distilled water outside and 40% sucrose in the bag. So that's our independent variable. Look at two different values of concentration gradient across the membrane. And then dependent variable is just rate of osmosis. How are we actually going to measure the rate of osmosis?
By how much water is diffused into the little thing, like time, like we're going to time how long it takes how much water to diffuse into it.
But how are we going to know how much water is moved into it?
We will measure the difference with the initial volume of water and the final volume of water, and with the difference.
How are you going to, are we actually measuring volumes? What are we actually measuring?
Millimeters.
Millimeters. So describe more how this thing works. How are we actually going to measure? What's that?
So based on like what the lab says, we're going to have like the little little thing of water with the dialysis film underneath it, and then it'll have like a pipette, and we'll measure how far up the pipette the water climbs, and we'll start with like a 0 millimeter.
Yeah, so that is our actual measure, right? I mean, so this is important. Hopefully, in addition, I mean, part of what you've hopefully done in order to do your pre-lab was actually read the procedure. So in order to think about this, we have the osmometer, which is basically, let me do narrower thing here. We basically have, it's called a Thistle Tube. It's a little plastic thing like this. We're going to have our dialysis membrane kind of on the bottom here. Dialysis membrane. This is a membrane. It's, in this case, it's artificial. It's this kind of plastic, and it has the ability to let water cross. So water can cross. But not sucrose. And other large molecules also get trapped. And we're going to talk about what dialysis is later. Dialysis is actually a kind of the sister to osmosis. In osmosis, you think about solutes are trapped and which way is water moving. In dialysis, you actually think about things, a semi-permeable membrane, but in terms of the things that can and can't cross the membrane itself. So dialysis actually is stuff, thinking about stuff that can cross a membrane. So we don't want to, don't get too confused there now. But this membrane here is constructed so that sucrose cannot pass, but water can pass. So any sucrose that's in here can't leave through that membrane. And we're going to put this in a beaker. It's just distilled water in it. And then we have a little tube that is attached here. So if all else being equal, and we usually dye the water so you can see if it's moving. All else being equal, if there was no concentration difference between the inside of this little thistle tube and the water in the beaker here, what would the level of the water in the tube be?
The same.
The same.
The same, because there's no net pressure in or out, right? So all else being equal, the level of water in this tube is just going to be the same level as the water in the beaker. What if I do have osmosis? So let's say I have sugar in here. Sugar can't leave. So that means the osmolarity in here is now some non-zero number. The osmolarity out here is still going to be 0, right? Because there's nothing dissolved there. Which way is the water going to move?
Into the tubing.
It's going to go move in. And as it moves in, what are we going to see?
Dilution of the color concentration.
What's that?
Would the color, like the dye concentration, get diluted as water?
Yeah, you're not really going to see that.
Okay.
What are you going to actually physically see, be able to measure to tell that water's actually moving, crossing in?
The water will raise.
Up the tube.
The water
Will raise
start
up.
moving up the tube, right? The more water comes in, the higher it's going to go up the tube. Does that make sense? This is going to be how we are going to actually, in a practical way, measure which direction is water moving and how fast is it moving. You know, so we can measure what is the height of this at 10 minutes. What is the height at 20 minutes? What's the height at 30 minutes? And then we can see the difference in height per unit time. That's going to give us a rate. Remember rate? It's just distance over time. So we can say how many millimeters per minute is the water moving up the tube. That's going to give us our dependent variable. We want this rate of osmosis. You know, what we're actually going to be able to measure is millimeters per minute that the water is moving up the tube. Right? Do y'all see that? So that's going to be, this is going to be the actual measured dependent variable. Even though we really are interested in rate of osmosis, what we're really going to be able to measure is the rate water is going up the tube, and that we are saying is, you know, going to be, you know, related, or, you know, our measure of rate of osmosis. Does that make sense? It's important to kind of understand the basic setup here before we get deeper in. What evidence are we going to have? Like here I am saying, you know, trust me, water can cross, but sucrose can't. How do you know that I'm not just making that up? COVID's not going to kill you. Don't wear a mask. It's fine.
Well, the instruction in the lab notebook says, lab manual, that specifically says that sucrose won't be able to cross, only water
Right.
will
But
cross.
that's just more words. What do you have in the real world that you can use as a data point to know that, you know, I wrote the lab book, you know?
Sucrose are like, they are glucose and a fructose, like a two disaccharide, like a larger molecule. Then
And so
water won't be able.
let's come back just
Well,
to
if sucrose can cross the membrane, the whole experiment's not going to work. It's just going
And
to stay
isn't
level.
the sucrose dyed so you'd be able to see if the other water changed color?
Don't worry about the color. The color is irrelevant. What is the core thing that I know that I can?
If sucrose can cross the membrane, then you're not going to have water move up the tube because there's not going to be osmotic pressure.
Right. So what's going to happen instead? If, let's say I put my 20% sucrose in my thing here, or let's say 40%, because I just accidentally wrote 40%. And let's say sucrose could cross the membrane. What would happen?
Sucrose would diffuse.
Yeah, sucrose would leave. It's like no sucrose here, 40% super sugary here. The sucrose is just going to leave until it's equilibrated inside and outside, and there's no osmotic gradient.
There's going to be an isotonic gradient, like concentration. I mean, isotonic, like equally.
Yeah, I mean, ultimately, it's going to diffuse out until it's just the same inside and outside, so then there's no osmotic pressure at all.
It wouldn't be very good lab etiquette, but you could just do a taste test on the DI water.
No, but what I want you to be thinking about here is if the water is actually up the tube, that's not going to, especially if we let it sit, if we hit some equilibrium, we walk away and we come back and it's going to settle in and we say, well, the water is up the tube compared to the level in the beaker. I know that there's got to be some pressure pushing the water up the tube. But there's got to be some osmosis going on. If sugar could cross the membrane, it would just cross and leave and equilibrate, and therefore there'd be no osmotic pressure. Does that make sense? Will the water just keep going up the tube forever?
No.
No, why is that?
Because at some point there's going to be no net movement.
But why? What would stop it?
They're going to be in equilibrium. It's going to be,
Same osmolarity.
it's going to be the, yes.
I mean, osmolarity, if you think about it, it's 0 here, nothing. Inside here, there's always going to be some sugar. It's going to be less and less sugar, but it's always going to be non-zero. It's always going to be some sugar dissolved in however much water is in there. So osmosis for eternity is always going to have some net movement of water into the membrane just due to pure osmosis, right? Right.
The Turgor pressure of the solution is going to equal the atmospheric pressure.
So this is where you've got to think about gravity. As the water goes up the tube, we create what's called a hydrostatic head. Basically, the gravity pulling the water back down. Basically, the water pressure. So we have this water pressure pushing down. And then the osmosis, kind of this osmotic pressure pushing up. You know, and when those balance out, then we're going to hit some equilibrium where there's no net movement anymore up or down. Right? The higher it goes, the more gravity is trying to pull it down. So at some point, you're going to hit some place where they balance out. In the beginning, there's no hydrostatic head. The water is just at the level of the beaker, and osmosis is winning as it goes up. But the higher it goes, the more the gravity is pulling it down. So at some point, you're going to hit a balance. Does that make sense? I mean, what if my tube was horizontal rather than vertical? How long would this experiment go on?
Forever.
It'd go on pretty much forever. It would go slower and slower and slower as the water in here became more and more and more dilute. But the reason it doesn't go forever upward is because gravity. I mean, assuming that the tube is tall enough. If the tube isn't tall enough, you know, it's just going to kind of spooge out the top, you know, before gravity builds up enough force to pull it back and counteract the osmotic pressure. So when you're thinking about, like, so what if I had my, again, here's my distilled water, and here's like 40% sucrose. You know, the osmosis coming in, it's going up, it's going to hit some level where it finally balances the gravity pulling down and the osmotic pressure. In fact, when we talk about the word osmotic pressure, that pressure is how hard you have to push down to just balance and equal it out. So osmotic pressure, water's coming into osmosis, hydrostatic head, the water pressure pushing down due to gravity. What would happen? What if I, instead of distilled water here, I had 20% sucrose outside? What would happen now to the system?
The water level in the tubing would go down and go into the beaker.
Yeah, so what's happening to the gradient here? What happened if I, instead of distilled water, I now have 20% sucrose outside? What is the gradient, the difference across the membrane like?
It's smaller.
It's smaller, which means less osmotic pressure, which means you're not going to get up as high, up high the tube before gravity can balance you out. Does that make sense? I want to make sure everybody kind of starts getting a feel for what exactly is going on, what we're seeing, how to interpret our results before we actually kind of dig into it more formally. So are there any questions about this? Does it make sense?
What's osmotic pressure again?
This is the pressure that's basically you can come up with, you can quantify it by saying how hard, what force do you need to push down to stop the water from coming in? So this is, it's the force due to osmosis, and to quantify it is like push against it, and however hard you have to push to exactly balance it, that's the osmotic pressure.
So it's the gravity.
In this case, it's gravity.
Okay.
So, crystal clear?
Could you please explain one more time this this particular condition with the 20% sucrose outside and 40% sugar out inside of the funnel?
All right, so again, the main thing we're looking at right now that affects the rate of osmosis is the difference in concentration across the membrane of sucrose in this case. So if we look at the difference between 0 and 40% in our case number 1, and compare it to the difference between 20 and 40 in case number 2, you know, case number 1 has a it's got, you know, twice as high of a difference. So this is a much higher gradient than this, which means we're going to have a higher osmotic pressure in this case than this one, which means the amount of pressure pushing up is going to take us much higher and condition 1 before gravity balances compared to condition 2?
Professor, the pressure that the water on the tube makes against the osmotic pressure is the hydrostatic pressure?
Yes. It's
Okay.
basically the water pressure pushing down.
Okay.
So
Down.
osmotic pressure is pushing up then. I thought it was gravity.
I mean, it's the hydrostatic head is due to gravity. It's due to the weight of the water coming, pushing back down the tube.
But then I thought you said osmotic pressure is gravity pushing down?
No. Osmotic pressure is the pressure due to osmosis going up.
Okay.
It has to, and it exactly, when this thing is in equilibrium, it's exactly balanced by the force of the weight of the water pushing down.
And that's
Okay.
hydrostatic pressure.
That's the hydrostatic head. Yeah.
Okay.
So the osmotic pressure is pushing up. Okay.
Yeah, the osmotic pressure is pushing up and the hydrostatic head due to gravity is pushing down. So, actually setting this thing up, it can be tricky. You got to really make sure you seal the little membrane on this thing really well so it doesn't leak. And normally what we do is I split you into 2 different groups, and half of you would set up your things with 40% in the thing. Half of you would set these things up with 20%. And, there's lots of sources. What are sources of variability in this thing?
Temperature?
Temperature, for sure.
Concentration of solute.
So the concentration of solute, and we're trying to control that, right? Make sure we don't contaminate the solutions, that they're well prepared, that we don't have like anything dissolved in our, that's why we use distilled water. When I write DH2O, that means it's pure water. Like when you pour water out of a tap, so this is not equal to tap water. Tap water has all sorts of minerals dissolved in it, different ions, different things that help kill the microbes and stuff, so it's potable. So, it's part of the reason, like, tap water conducts electricity better than distilled water because it's usually got dissolved minerals. When we talk about hard water and stuff, it's got all sorts of dissolved minerals in it, right? If you have water, you've probably noticed that water often like ends up having these kind of crystalline like deposits on things after a while. You start depositing, calcium carbonate and things like that. So we're going to use distilled water, and that's to control for the solutes. Make sure that outside it really is 0 osmolar, because we use distilled water, which has got nothing in it. Other sources of variability.
Barometric pressure.
Yeah, barometric pressure, although that's going to be kind of equal on either side. So we need to make sure that we don't like have anybody, blowing in here on the top or something.
Dr. Permeability of the membrane, porosity of the membrane.
Yeah, so making sure that we're using the same kind of membrane and trying as much as possible to stretch it out the same way. Making sure there's no tears in there that make it easy for stuff to cross. There's all sorts of little things like that. So we're going to do now, we're going to do this thing, doing the Osmometer Lab. The first thing is going to be, you just get to sit back and relax, and you get to watch me kind of assemble everything for 9 minutes, just so you can actually, you're going to be interpreting the data, but it is worth actually seeing how the data was actually acquired. because in your discussion, that's where you're going to be able to talk about things you trust or don't trust, or things that might have gone wrong, or things, you know, this and that. And then we'll actually, you know, show you like what the results look like. And, you know, and it's interesting. I can stop this sharing. I was talking with one of my friends this weekend, you know, about, you know, our situation of having to watch the labs being done, but, you know, getting to at least analyze the data. And, you know, at some level, it's, you know, it's disappointing. Not at some level, at many levels, it's disappointing. But, you know, one of the things he was, so this is a friend who's a senior scientist at Genentech now. And he was thinking about his experience of his initial science classes in college. And he said, you know, one of the most important things that he did get out of his labs, part of it was technique, but a lot of it was how to think about interpreting your data. What does it mean? He talked about one of his labs where he spent like the whole night just trying to figure out how to make sense of this, and looking at it this way, looking at it this way, and coming up with maybe it means this, and I'll write it up like this. And, you know, finally, like coming into class the next time and, you know, here, I think maybe this is happening, but really I'm like really kind of frustrated because I couldn't really figure out like a really nice story to tell with the data. And his professor is like, you know, that's because there's nothing to tell. You know, the data isn't actually, the data wasn't telling what you were hoping. It doesn't really say anything interesting. And he was realizing it is something he taken to his career, like as a professional scientist out there in the world, is just realizing, you know, there's a lot of subtlety in how you make sense of what you're seeing, what you're observing. And also sometimes realizing sometimes it doesn't say anything, other times, sometimes it is saying something, but not what you expected. You know, so all of the stuff you're doing is actually really helpful and important skills, like the stuff with the jumping lab. You know, you're probably going to find some things where you find interesting results. Sometimes you're going to find opposite results. Sometimes you're going to find, I was really expecting there to be a result, but it all looks like random noise. And all of that is really important experience. Even if you're not having the physical experience of doing some of these, just the experience of working with the data and trying to figure out how to interpret things is a really important skill that we will be kind of developing, you know, throughout the semester here. So just, it was actually helpful for me to hear him like, you know, actually a lot of the really important stuff I got out of the lab was this other, you know, this, you know, so it made me feel a little better about, you know, yes, you don't get as much of the hands-on, and a lot of that you will get in your subsequent classes, particularly the more clinical stuff. So what we're going to be focusing a lot on in this lab is, you know, how to responsibly interpret your data and how it relates to the concepts that we're talking about. Before we actually show this, one last thing. So people had their hypotheses. Hypotheses. What were some hypotheses for what we expect to find?
The greater the concentration gradient, the faster the rate of osmosis.
So greater, and then given the fact that we have 40% versus 20% in the bag, which one is going to go faster?
The 20%.
40%.
40.
Right, we got 40 versus 0 and 20 versus 0. Which one has a higher gradient? Which one has a higher difference across?
40.
Yeah, this is like twice as high a difference, twice the high a gradient as this. So which one of these is going to go up the tube faster?
40%.
Yeah, this one's going to go probably up twice as fast as this one, right? So that is, hopefully that makes sense. That feels relatively intuitive to y'all. If I want to take it one step further and look at time versus rate. So as time goes on, so I could say, let's say this, the 40% is gonna, when we first start, it's gonna be some value. The 20% is probably gonna be quite a bit lower, right? Because that's where we start with the initial conditions. Water is coming into both of them due to osmosis, but it's got twice as high a gradient in the 40% as in the 20%. Maybe I'll make this blue here. So I'm going to have my 20% rate is going to be quite a bit lower than the 40% rate. What's going to happen as time goes on? What's going to happen to these rates?
It's going to slow down as they reach equilibrium.
It's going to slow down. What two things are happening to make them slow down?
A lower concentration gradient.
So one is a lower concentration gradient. The more that water comes
head
in,
is getting heavier.
and the hydrostatic head. So we have both diluting the concentration in here, so there's a lower osmotic pressure, plus increasing the level of this column of water means there's more gravity pushing down. So both of those things together are causing this to get slower and slower as time goes on until we hit some equilibrium where there's no movement at all. We're going to probably expect this one to be, also going down, but always going to be lower than the other one. Does that make sense?
Mm-hmm.
So this is kind of theoretical. We might or might not see that when we look at the actual data. Again, what kind of other sources of variability that can mess with this?
Measuring with the ruler might be
measuring, just measuring errors. Measuring errors can be a big problem. Depending how tight this thing, it could be the little thing like pooches out. Like it might be it's filling with more water, but this thing is becoming kind of more distended. So instead of water going up the tube, we're just getting this thing blowing up a little bit more like a balloon like on the bottom or something. So there's lots of things that can go wrong that might not make our final graph look exactly like this. All right. So now it's time to kick off your shoes, sit back and relax as you get to see me in the lab this summer with Tina filming me, setting up the Osmometer Lab. So here we go. Yeah, I have to say, it's like one of the few very fun things about this whole shelter in place. Filming the labs actually was kind of fun. Let's see, because you actually got to like do something that wasn't just sitting in front of a computer. All right, here we go. All right, so this is the Osmometer Lab. And for this lab, we're going to be measuring the rate of osmosis under different conditions. And to do that, we're going to need an instrument called an osmometer. So the osmometer is actually super simple. Osmosis. Osmosis is just diffusion of water across a semi-permeable membrane. So if we're going to have osmosis going on, we need a semi-permeable membrane. That's this stuff right here. This is called dialysis tubing. Actually, it looks like tape, but if you kind of massage it between your fingers, it actually opens up and becomes a tube. And then we're actually gonna take this tube and we can like snip it and cross the bottom to make a smaller part and up the side so we can kind of open it up. And basically now I have a sheet of this dialysis membrane. And this dialysis membrane is basically a plastic that has tiny little pores in it that will allow water to freely pass, but will block the passage of large molecules such as sucrose, which is going to be the thing we're looking at today. We're going to have different concentrations of sucrose solution, which is sugar water. And with the sugar water in this membrane, the water can move across from one side to the other, but the sugar molecules are trapped on whichever side they're at already. So we're going to take this. I have kind of a prettier one that I already cut up here. And I've got my little square of dialysis membrane. Then the other part of this is going to be a thistle tube. Thistle, I'm not even sure why it's called a thistle tube. I guess it looks like a thistle. It's basically a tube with this little funnel on top. And we're going to actually use it upside down. And take this dialysis membrane, put it here. We're going to then be able to have one solution inside the tube on one side of this membrane. And then we're going to actually take this whole thing and put it in a beaker of water. So we're going to have distilled water on the outside of the membrane. We're going to actually have our sugar water inside this little funnel on the inside part of the membrane, which is going to create a situation for osmosis. We're going to have. And I just want to reiterate, the colors are purely just so you can see stuff going up the tube. You want to kind of ignore them in terms of osmosis or particles or anything like that. The color is purely to be able to more easily visualize things and to make it easy to see which is the 40, which is the 20%. But the concentration of the dye molecules is so little, it's basically irrelevant in terms of osmosis. So don't think of the dye as something that's part of the experiment other than to make it pretty like this.
Okay.
A gradient, an osmotic gradient, right? We're going to have concentration of sucrose on one side, a different concentration, in fact, zero concentration, distilled water, zero concentration. And that's going to drive osmosis, movement of water across the membrane from the beaker into this little thistle tube, which is going to start going up and up and up. So we're going to be able to actually monitor the progression of the osmosis by seeing how far up the tube this sugar water goes as more and more water comes in to add into that. So I'm going to put this thing together. I take my tube. I need to really seal this dialysis membrane really tight because I want to make sure any water that comes in is going across here and nothing leaks out. I'm going to pull it really tight like a drum head. Boom, 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 boom. And I'm going to use not one, but a few rubber bands. As this experiment proceeds, the pressures actually get pretty intense. So any weakness is going to end up with having water and stuff leak and sugar leak out where it shouldn't. So I am basically right now pulling this membrane really tight, making sure that it's right up against the lip, making sure that there's no leakage points. I'm going to take another rubber band and I'm going to put it around, oops, again. I can't overemphasize how easy it is to have this experiment fail if you do not really seal this really well. So there's two. I think this is probably enough. You could even do a third if you want. Now I need to put my sugar solution in here, and that's easy to just do with the pipette. Our old friend, the pipette. Here, the volume action is more just an instrument to get it into the little hole here. You can suck it up. As you put the tip of the pipette in here, you don't want to push it all the way down because you're going to basically create a vacuum and you're actually going to not be able to have the sugar water go in because the air is going to block it. So I'm going to start filling this thing. Whoops. Do. And do. If I take them one more time. Do. Stop singing. All right, so this is filled up, and I should mention that color is purely just for visibility. It doesn't affect osmolarity or the concentrations. It's purely just for color. The red and the blue, don't think of them as players in what is the osmotic rate going to be. It's just so you can see things. Then what we need to do is put the tube into this funnel. We want to make sure, again, the sealing, making sure there's no leaks is really important. So for this, I actually have Teflon tape, which is used for plumbing. If you haven't seen it before, it's incredibly useful for lots of household tasks, but it's really useful for osmometers. You basically wrap it around a bit, and you kind of pull, and it rips off. Make sure you haven't blocked the hole. And this is going to create this really nice tight seal when I kind of jam it in here. So again, in this case, we have 40% inside distilled water in our beaker. So, again, our independent variable here, what's changing? We've got, in this case, the thing that's driving osmosis is this diffusion, the concentration gradient, the concentration difference across the two sides of the membrane. Here it's 40% in the funnel, 0 because it's distilled water in the beaker. The other group where they fill with this 20% solution, they have a different concentration gradient across the membrane. And we'll see how having a different concentration gradient across the membrane affects the rate of osmosis. Now I'm sticking this in here, and that's what I want to see. So at time equals 0, you can see the water is at this level up the tube. And because that's basically my time equals 0, I want to basically tape this onto the ruler such that any amount of water that crosses the membrane and enters into the Thistle Tube, which is going to drive the movement of the water up the tube, will be super easy to measure because it's 0 right now. And as it goes up the tube, I can just see directly off of here. In fact, I probably want to push it over a little bit because then it's even easier to see because it's against the millimeters. So that's going to make it really easy to, again, in rate, as Tina was saying earlier, rate is going to be the distance divided by the time. So for any 10-minute segment, you can measure a rate for actually that particular 10-minute segment. You can say, so in that 10 minutes, how far did it move? And divide that by 10 minutes, and it can give you millimeters per minute. So while you guys go and go take a little break, I'm going to now set up the osmometer with the 20%. All right. So sharing. So are there any questions about that? It kind of gives you a nice feel for how this thing actually is put together and what that dialysis membrane really looks like and what it is. In reality, when we do this in person, part of the reason why we do multiple groups is often somebody has one that leaks. Because it's tricky to get it just perfect. And that's part of science too, is sometimes getting a good experiment can be tricky to put together, even if you know what you're trying to do. So what we're gonna do now is just show you the results. And that's just gonna be a few minutes. So I'm going to, all right, here. Whoa, hold on. I need to figure out what's going on here. There it goes. All right, I'm going to. All right, so I'm going to go back to sharing the screen again. All right, so this is just 3 minutes, and this is where you see what it looks like when they're both going. All right, so now we have these two osmometers set up. And this one, they're identical except for the solution that is inside the actual little funnel. This has the 40% sucrose. This has the 20% sucrose. And again, that sets up a condition where we have two different concentration gradients across that dialysis membrane. In this case, it's 40% on the inside, 0 on the outside. In this case, 20% on the inside, 0 on the outside. So we have like twice as strong of a gradient. We're going to see how that affects osmotic rate. And again, that's going to be reflected as water enters in. It's going to start rising up. These are both at 0 right now. And I've just set my timer here. So at 10 minutes, I'm going to be able to see the height in both of these. At 20 minutes, at 30 minutes, at 40 minutes, at 50. All right, so it's 10 minutes, and now I want to just do my readings here. And again, here I can see the 40% has gone up quite a bit, actually. We're about 1.8 centimeters or 18 millimeters above 0. Remember, it was at 0 when we were at time equals 0. Over here, we are much less height gained. We're only about, I'd say like 8.5 millimeters. So, you know, I write that down in my data. I actually have my lab book open, put that down, and then keep the experiment going and take the next data points at 20 minutes, and again at 30 minutes, and at 40 minutes. And that's the data that you're going to be seeing in the spreadsheet that we supplied for you is, you know, groups, you know, more than one group, because doing one experiment once doesn't really tell you really if it's a consistent result or maybe it's like just a one-off thing that happened. But if you do the same experiment again and again and you get similar results, or you have many people doing the same experiment all at the same time and all getting similar results, then you can start believing that your results are actually telling you something important. So, you know, we wouldn't just, you know, this would be suggestive if we saw these two results and compared them. But if we had a number of different groups all doing this at the same time and looked at the trends across all those groups, and if there were similar things happening, you know, then you start getting rid of the noise and looking in at the true processes that are underneath it all. All right, so right, and so the data is just here. So if you like, these are those videos, and then here, you know, Osmosis Lab from spring 2020. Because the spring people, we didn't shelter in place till halfway through the semester, so they actually got to do this one. And I'm going to and the data, it looks like this. Here, the 20% sucrose group, there's a 40% sucrose. What? Hold on, something went wrong. Share screen, get it back. So 20%, 40% sucrose group. Again, there's 3 groups doing one, 3 groups doing the other. And if you take a look, they're not all exactly the same, but they all have similar trends. If you look in general, the 40% sucrose group, they're not the same, but they are all appreciably higher than the 3 groups that were the 20% sucrose group. So, and this is why you don't trust just doing an experiment once. It usually doesn't turn out exactly the same. But if you do the same thing again and again, it's kind of like when we measured like jump height or something. You start getting things that are clustered around the real thing, and you average them and you can get a sense of what's really happening. Most of these graphs that we have for you, or most of these spreadsheets, have multiple pages. Down here at the bottom, there are these little file folder tabs. So I have like a height graph here. This is, so you can actually just use this. You could print this out rather than having to generate it yourself. And it's basically the height up the tube, and it's obvious they both start at 0. That's what we set it at. We made sure it started at 0. When we look over time, this is kind of what we'd expect. It's like we'd be, over time, they're both going up. It's going up the tube. We obviously did not hit that equilibrium at 40 minutes because both of them were still rising. And at any point in time, we can see that more water has come into the 40% than the 20% because the rate was always faster in the 40% than in the 20%. So this is, we look at this, do kind of our sanity check, and it's like, this makes sense. This is not making us worried that something really weird is going on. This is kind of what we were hoping to see. Again, height was not our actual dependent variable. What was our dependent variable?
The rate.
The rate.
So to calculate rate is just distance divided by time. So basically for each of these 10-minute things, we can just take what is the height difference from like 10 minutes to 20 minutes and divide by 10 minutes. And that gives us millimeters per minute that the water was going in for that 10-minute block on average. So that's what this rate graph is, this next little tab here. This is showing for every 10-minute block, take that 10 minutes of height difference and then divide by 10 minutes. And for the 40%, we see a lot, it's pretty similar to what we were predicting. Starts high and gets lower in general along the way. 20% is starting much slower. There's this weird hiccup here. Who knows what it is? In your discussion, you can discuss what the heck do you think happened there? I'm sure it's one of the sources of variability probably that is responsible for that. That's normal. It's normal that your data has some weirdness that, it might be real or it might not be real. A lot of times it's not real. A lot of times it's just, the term we use for things that show up in your data but actually aren't related to a real effect is an artifact. So that word artifact shows up. Maybe I should just write it in. I can use my little annotator. When you see something like that, that is likely artifact. And when we're talking about scientific data, an artifact is something that happened spuriously or randomly and wasn't actually due to something real that was happening in the process. So that word shows up a lot, like, that's not real, that's just an artifact. So, all right, so that is kind of what you're going to be working with in your discussion for this lab. You'll be using the, your understanding of the process and your understanding of, the experiment and the methodology, and then this data. We have the height versus time, the rate versus time, and you'll discuss what happened and how it does it support your hypothesis or not, and that's the osmometer part of the lab. Are there any questions about that? There's one other thing on this data sheet, this auger diffusion, which we're going to talk about this now. So just when you're wondering where is the data for the auger diffusion, it's in the same data sheet. It's just there's the osmometer data, raw data, the auger diffusion raw data. So we'll come back. We're going to discuss this now. All right, we're going to stop sharing. So before we leave osmometer, any questions regarding osmometer? Okay. So the other part of the lab is demonstrations of diffusion and osmosis. Demonstrations of diffusion, actually, not of osmosis in reality. Oh, and I need to, actually, I think hold on a second. This is interesting. Give me 1 second here. Okay.
Now, right now, it's fluid.
Okay.
But let's increase the nitrification a bit more.
Okay, so I did find, okay, so there's two parts to this thing. What is the first part of the demos?
Brownian motion.
Yes, Brownian motion. So this, again, this one is cool to see in real life, but it's still worth understanding the basic idea here and watching a little video of it. So we talked about what is actually driving diffusion. Why do things diffuse from high to low concentration? If it's passive, which means we're not adding extra energy to it, how are things moving?
Because of the molecules collide.
Yeah, it's because molecules are just jiggling. Anything that's not at absolute zero, the molecules are not just sitting there quietly waiting to get bumped or something. They have, we talked about translational energy, moving from one side to another, vibrating and rotating and doing all this stuff. And that's what's ultimately driving diffusion, is all of this motion. Can you actually look under a microscope and see the atoms and molecules jiggling and moving? No, I see everybody's head going. That's good to see. Light microscopy is very limited at what you can actually see. But what you can see is evidence that this is happening. And the way we do it, one of the classic things is observing Brownian motion. And the basic idea is, usually you have like a microscope slide and you put a drop of water on there. And then you put something really, really small on it, really small and light. In this lab, we use these tiny little crystals of Carmen dye, which you could think of like little red rocks. They're easy to see, but they're dinky. When I was like a kid, I remember the first time I ever saw Brownian motion was with pollen grains sitting on a drop of water. And the basic idea is here, we have water, and the water is in constant motion. Whoops, I want to get, so here's the water. And these things are floating in the water, but the water is, if we like zoom this up into like the viewpoint of like Maxwell's demon, something that it can actually see atoms and molecules. You can't actually see this, but let's say my tiny little crystal is like this, and I have my little H2Os around it. Each of these little H2Os are in motion and whacking off the side of this thing. So imagine if you were standing in the middle of the room and there were people all around you shooting you with tiny little BBs or something. And you keep getting like, whoa, 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 back. Wherever you get hit, you're getting, right? So you are gonna be jiggling. You're not really gonna be moving much of anywhere, but you're not gonna stand still because you keep getting like hit from one side or the other. And somebody who's standing back and watching you are just gonna think you're like kind of having a bit of a seizure or something. They're not gonna actually see anything's actually hitting you, but they'll see you jiggling, so they'll know something's going on. So that's what Brownian motion is. Brownian motion is you look at really dinky little things in water under really high magnification. So when we do this in lab for real, you get up to 100 times, you don't see anything. The little red rocks, the little Carmen dye, they just look like they're sitting there. By the time you get up to 400 times magnification, they get all jittery. And it's cool. You can, and they're not, they look alive. They look like they're little living things going zzz, but you realize they're just little crystals getting whacked by the water molecules that are invisible. You don't see. So I'm gonna share, this is, and I'm gonna link to this thing a little later. I realize I didn't link it yet in the module, but I will put it in there. So here's this little thing of observing Brownian motion.
You actually see some Brownian motion. And there we go. So I adjust my magnification. I can see my little drops, and they're all jiggling around. And that jiggling around that you see is Brownian motion. So now your job is to pick a good candidate bead to trace its motion and measure how far it travels in given lengths of time.
So you don't have to do that. I mean, and it's subtle. In this video, it's kind of harder to see quite how magical it really looks. And I'll do a little bit more searching to see if I find another example. But when you look at it, they really look like they're these weird little jiggly living things. And it's important. That's when you get into microbiology, it's actually important to understand this. You know, if you're looking at something, just because it's tiny and jiggling at high magnification doesn't mean it's alive. It can be inanimate, just getting jiggled by Brownian motion. What would happen if you heated this up? How would that affect the Brownian motion?
There would be more movement.
Move faster.
It'd be more movement, kind of faster and probably larger distances because you've got more energy worth of the water molecules whacking into everything. So that is that. The other thing we do to kind of, we talked about molecular weight versus diffusion rate, which rhymes. Molecular weight versus diffusion rate. What was the relationship here?
Inverse.
It was inverse. And we talked about larger things with the same amount of energy are kind of going slower. Smaller things with the same amount of energy tend to be zipping along faster. For the lab, we're going to use two things that are also kind of pretty colors that have different weights. We're going to use potassium permanganate and methylene blue. Normally, I would say be really careful with them because they'll stain your clothes, but that's one of the benefits of doing this online is you're not going to stain anything. Methylene blue has a molecular weight is 2 times the molecular weight of potassium permanganate. It's much heavier. So what is your thought in terms of, if I was just going to look at pure diffusion of methylene blue versus potassium permanganate, which is purple, which one do you think is going to diffuse faster and which is slower?
Potassium permanganate will move or diffuse faster.
Exactly. You know, and the way we're going to look for this is we have a little auger plate. So most of you probably know what auger is. It's like Jello. It's like Jello. And we're going to make a little well in the auger plate and then fill it with our dye. And then watch over time to see how far it diffuses out into the auger. So this distance here, how far has it gone after a half hour, after an hour? So this is going to be our way to measure the rate of diffusion of this methylene blue solution or potassium permanganate solution, is just to see how far it goes. Why am I doing it in auger? Why don't I just put it in some water and measure the diffusion?
It would mix together if it was in water.
Yeah, so this is, so if you think about, let's say you're sitting here in the room and someone comes in, I mean, I don't know if it's even appropriate anymore. Let's say it's your little old lady, the old classic little old lady who is wearing a lot of perfume. You know, and this is something we're going to talk about your senses. As you get older, almost everything gets worse, including your sense of smell. So it's actually really common for people to put on way more perfume than you think they need because they don't actually realize how much perfume they put on. So she walks in and you're like, whoa, she's got a lot of perfume. Is that diffusion? How is the perfume molecules getting over to your nose over here?
By air.
So
By air molecule.
So it's by air, and how is it moving through the air? So it's actually, it's not diffusion. So diffusion, we're going to see diffusion is really good for things moving over a fraction of a millimeter. But once you get farther than that, it's not going very far at all. If you were going to use pure diffusion and have something diffuse like a 10 centimeters, you'd have to wait months. So when you're smelling this perfume, what you're really smelling is the air currents. Right? If you like take a drag off a cigarette and you'll watch all the pretty smoke like wisps flying through the air. That gives you a sense of there's all these different currents in the air. You know, air is constantly moving around in these little currents. That's what's carrying the smell of her perfume over to you over here. When you are putting sugar in your cup of coffee, I got my cup of coffee here, and I put in some sugar. Do I wait for that sugar to just find its way to distribute evenly through the coffee? It would take forever. It would take, again, it would actually take probably like a month if you wanted pure diffusion to have the sugar equally distributed everywhere. And instead what you do is you put a spoon in there and you mix it around. Again, convection, you got the currents. So when you want to move things a far distance, you don't use diffusion. Like how do we actually get the oxygen to the cells in our body?
Through our blood.
Through the blood, exactly. So when we want to actually, you know, basically in your lungs, we have air, and then there is diffusion across a fraction of a millimeter to go from the alveoli into the bloodstream. And then whoosh, bloodstream, convection, flow. And then once we get to the destination, there's the cells where we're going to actually deliver the oxygen. Then again, a fraction of a millimeter of diffusion where the oxygen leaves the blood and goes into the tissue to the cells that it needs to service. But when you want to move something a far distance, you need flow. You don't use diffusion. Diffusion is only effective over a very short distance. So that's important. That's something you're going to see in this thing I am about to share with you now, which is Tina showing the behavior of the two dyes in the agar. So we're going to go back to the module. And now, under this doing the diffusion demos, under lab 3B here, here is Tina illustrating this.
All right, so here we're going to do the lab that is a demonstration of diffusion, and the experiment I'm setting up here is the one where we're going to put some of these dyes, methylene blue and potassium permanganate, into these wells of these agar plates. So the idea here is that the molecular size is much larger of the methylene blue than the potassium permanganate. And so we want to see how that will affect the diffusion and diffusion rate in this agar plate. So I'm going to fill up the well with each of these dyes, and then every half hour we're going to come back and measure how far it has diffused through the agar. Make sure, and you might have to come back and just make sure that it's topped off. So if it goes too low, you might want to add a few more drops. And we're also going to do our potassium permanganate. And when you measure after a half hour, of course, we're going to use the metric system. So we're going to use the millimeters or centimeters. And each time you want to measure from the edge of the well out to the edge of wherever the dye has diffused out to. So of course this is time 0 and it hasn't diffused at all. So our distance is going to be 0, and then we'll come back in a half hour. Okay, so I'm back here at our diffusion experiment, and it's been a half hour, so it's time for us to measure how far the dyes have diffused away from the well edge. And so if I start here with the potassium permanganate, I will use the metric side of this ruler, and we're going to measure it in millimeters. I'll line the 0 mark with the edge, and then I'm going to read the very edge here. It looks like 4 millimeters in 30 minutes, or 4 millimeters in a half an hour. And then when we go over and look at the methylene blue, we can see that it looks like it's diffused about 2 millimeters out in a half hour, or 2 millimeters in 30 minutes. But your lab manual is asking you to calculate the rate. And so when you have a rate, you want to divide the distance with time. And so you have a half hour here, but you're going to actually report it as millimeters per hour. Just like if you're driving down the freeway, you're going to be talking about your speed, which is a rate. It's how far you drove over some time. And so it doesn't matter if you're driving 2 hours or just 20 minutes, you're still going to report the rate as per hour. And so that's what you're going to do here too. Even though you just waited 30 minutes, we're going to report it as how many millimeters per hour did it diffuse in the first half hour. And then we're going to wait another half hour, and we're going to come back and take a look at how far did the dyes diffuse in the next half hour, and you're going to figure out the rate for that second half hour and see if the rate actually increased, decreased, or stayed the same. All right. All right, so we're here at our diffusion experiment, and we're going to do our time after 1 hour time point measurements. And so I'm just going to use the ruler again and measure from the edge of the well. And in this case, it looks like 7 millimeters for the potassium permanganate and 4 millimeters for the methylene blue. And so I also want to mention that we're using auger because if we were to put it into water, you would have convection forces mixing the dye around, and auger is a stationary molecular structure. So we're not going to get any convection, messing with our experiment. All right, so we're going to let this sit for another half hour and get our final reading after 1.5 hours. Back here for our last diffusion measurement. And so when we take a look at the potassium permanganate, it looks like only 7.5. And for our methylene blue, 4.5. So definitely it looks like the rate of diffusion is slowing down, but it's for you to calculate exactly what that rate is.
Okie dokie. All right, so there you can see, and again, I wrote, note that the recorded data for this part is contained as a page in the spreadsheet available in the previous Osmometer module. Here it is. Again, if I go new share. So again, this auger diffusion here. And again, if you look, potassium permanganate, the average distance that it's gone at any point is about twice as far. And if you calculate the rate by dividing how far did it go in that last half hour divided by 30 minutes, or by a half an hour if we're doing millimeters per hour, you can see that the potassium permanganate is going much faster than the methylene blue. You can also see, though, that it starts slowing down pretty dramatically. And I can say, just as a reality check for you all, like, we have left these things overnight, over many days even. At some, they don't even hit the end of the Petri dish. They start out kind of quick, but they slow down, and then they slow down more and more and more and more. So again, diffusion is good for going a short distance. It's how we're going to have neurotransmitters crossing the synapse. It's how we're going to have oxygen moving in and out of the bloodstream to the cells and stuff. But it's only good if you're thinking about things moving a fraction of a millimeter. It's not good for moving things a far distance. So are there questions about this part? All right. So for the in terms of actually writing this up, I actually do need to get you a better link to some Brownian motion, so you can, I do want you to watch some Brownian motion on a little video and kind of just describe it in your lab notebook, so you have a sense, like, what you see it. And then I also, in your writing, again, the discussion is kind of answering those questions that I have in the lab manual there. Like, what exactly are you seeing? I want you to understand why the little things are jiggling. So I want you to have in your discussion, make it clear that you understand why Brownian motion exists and what the implications are. In terms of the diffusion of the potassium permanganate and the methylene blue, it would actually be nice in your lab notebook to have a little, a little chart where you just have, like, potassium permanganate, methylene blue, 30 minutes, 60 minutes, 90 minutes. So you can have, and actually for each, wish I drew this bad. Let's just have it, let's say for potassium permanganate, at 30 minutes, at 60 minutes, at 90 minutes, and then millimeters, and then what is the rate? Average rate, I should say. Just so you have, so you can kind of make sure you understand what we were doing and where the data came from that's in the Excel spreadsheet. So the methylene blue would also be here. You'll also have a little chart at 30 minutes, 90 minutes, 30, 60, 90. I am not being very good here. Something like this. So you have for the methylene blue and for the potassium permanganate, just so you are clear about how do we get this data. Again, it's like we're looking at 30-minute progressive readings, measuring the distance that is traveled in each of those 30-minute blocks, and then calculating an average rate for that block just by what is the difference between how far did it move? It's going to be this minus this, or this minus this, divided by 30 minutes, or divided by a half an hour. If we're going to do millimeters per hour, it's going to be divided by a half, right? Because that's, you have to make sure your units match properly. So that is the demo part. there's no hypothesis or objective, but you should kind of describe what this means. You know, compare the methylene blue to the potassium permanganate, what you saw and why it makes sense from the theoretical framework we've been talking about. So that all kind of makes sense.
So is this going to be included in our osmolarity lab, like at the end of it, or in the middle of it, or is this separate?
So I would write up the osmolarity lab and then add this second after it.
Okay, and then do like a write-up of this part too, but we don't need a hypothesis or objective.
Correct.
Okay, thank you.
Okay. And I think the next lab is also just continuing these ideas of osmolarity and osmosis, except looking at actual living cells. So I'm going to, we have 10 minutes here. I'm going to just discuss very kind of briefly a little background on the terminology that you'll need, and also giving you the kind of core ideas you need to think about to develop your hypotheses, because you actually do need to have an objective and a hypothesis for Tuesday. Again, that's a long way from now. That's like, let's say Thursday, Friday, Saturday, Sunday, Monday. So Tuesday, you're going to have objective and hypothesis for the fluid balance and living cells lab. So next week's lab. Next week's lab, we're going to use actual blood cells. We used to use our own blood. We'd had the nurse, June, used to like just take the blood out of, we rotated, but now we buy cow blood, which is similar, but we'll talk about has some differences. So we have an RBC. RBC is just the red blood cell. What is the osmolarity of the stuff inside an RBC?
0.3.
Exactly. 0.3 osmolar. So in your hypotheses, you always got to remember that the cell is 0.3 osmolar inside. And then we're going to put our red blood cells in different kinds of solutions. So, and there's going to be different kinds of solutes dissolved in these solutions. There's actually a couple of independent variables in this lab. One independent, when we talk about the independent variables, one is going to be just the concentration of the solution that we're putting the blood cell in. But another is going to be the type of solute. The reason why we have to worry about the type of solute is some solutes can penetrate the membrane and some can't. So when we're thinking about osmosis, just like I talked about with the osmometer, it mattered that the sucrose couldn't cross the dialysis membrane. We really have to know whether or not the solute crosses this membrane on its own, or whether it's going to get stuck on one side or another and cause osmosis, contribute to the osmotic pressure. So independent variables. Can you see, is this bottom of the screen visible to y'all?
No.
No?
The yellow color is hard to see.
So we have independent variables. There's going to be solute type and the solution concentration. That clearer? You know, our dependent variable is going to be what happens to the cell. And there's a few things that can happen. Hemolysis, blood lysis, to break open. This is when the blood cells basically break open. We're going to be able to tell, you know, actually we have a nice, we're still okay, thinking about our assay for hemolysis. Assay meaning how are we going to tell if the cells exploded or not? We're going to have a test tube with some solution in it. We're going to put blood in there. When you mix the blood in, it's just going to all look red, right? If you take some red blood and you mix it in some solution and mix it up, it's all going to look red. But we talked about how blood is a suspension. The blood cells are actually big enough that gravity will pull them down. So I can take this thing, if the blood cells, you know, if the blood cells are intact, if I take this and I run it through a centrifuge, then the blood cells will all get pulled back to the bottom. And the top part, what we call the supernatant, is going to be a clear supernatant. Basically centrifuging, that's spinning super fast. And as you spin it super fast, you make gravity basically really strong. As long as the blood cells are intact, then they're just going to get all pulled down to the bottom. So this is evidence that there was no hemolysis. So if the blood cells are intact, there is no red left up in here after we spin it down. What if there is hemolysis? What are we going to see?
Little fragments up in the supernatant because they're going to be lighter weight.
So you're not going to actually see the fragments, but what you're going to see, each of the red blood cells is filled with about 250 million hemoglobin molecules. That's what gives them their red color. So this is looking red, but now it's not just red blood cells. It's actually just loose free hemoglobin, which are just molecules. It's no longer big cells with 250 million hemoglobin molecules. It's just hemoglobin in solution. And we take this through our centrifuge, spin it. You can't spin a molecule down. You can spin a cell down, but you can't spin a molecule down. So after we've spun it down, the supernatant is just going to stay red. So that is telling us that hemolysis happened. That means that the blood cells exploded, spilled their hemoglobin into solution, and therefore, when you spin it, everything just stays red. So we have like two possibilities here. You spin it, and the blood cells stay intact, in which case they all get pulled to the bottom and you see a red pellet at the bottom and the supernatant is clear. Or if the blood cells explode, all the hemoglobin goes into solution and stays in solution even after the spinning, and it looks red. Does that make sense? All right, so let's come back here. Let's start with a solute. Let's start with a non-penetrating solute. A non-penetrating solute, what are some of the things that we know will not penetrate through a cell membrane on its own?
A big polar molecule.
And give me an example. What's an example of a big polar molecule?
Glucose?
Glucose. Sure, glucose or sucrose, any of those sugars are big polar molecules. So if the solute in the tube is sugar, we can assume it's a non-penetrating solute. What's another type of non-penetrating solute that we've been talking about?
CO2?
No.
No. I'm so sorry. No. Wrong.
Wrong.
Super wrong.
What about the other thing we were looking at in the Chem Review lab?
Salt?
The salts. Salts are non-penetrating. We talked about how they have like that snowball that shows up around them. So unless you have a channel, ion does not cross. So salts are non-penetrating. So this would be like sugar or salt or something like that. And then we can talk about solution concentration. If the osmolarity is lower than 0.3 on the outside, I say it's hypotonic solution. Hypoosmotic solution. Hypo means less. So hypoosmotic solution means there's less dissolved outside than there is in the cell. So any osmolarity outside that's less than 0.3 would be hypoosmotic. If it's equal inside and outside, if it's 0.3 outside and 0.3 inside, this is called isoosmotic solution. Iso means the same. And finally, if I have more stuff dissolved out here, if this is greater than 0.3, this would be a hyperosmotic solution. We're in hyper, more. Hyperactive, more active. So hypoosmotic meaning less than 0.3 in the solution. Isoosmotic means exactly 0.3 in the solution. Hyperosmotic greater than 0.3 in the solution. What's going to happen? Again, assuming non-penetrating solute, what is going to happen in case number 1? If there's less stuff dissolved outside than in the cell?
It will shrivel.
The cell will swell.
The water's going to, remember this, the water moves in towards the solute, right? The water moves to where there's more stuff. Remember the solute kind of draws in, you can think of the water. So this, if the water comes in, we say it's a hypotonic solution. Hypotonic means H2O enters the cell. So tonicity is not exactly osmolarity, as we're going to see in a few moments. If we have the same osmolarity inside and outside, which way is the water going to go?
Nowhere.
It's going to be in equilibrium. It's not going to be net movement in or out, right? It's going to be what we call isotonic. Isotonic means water is in balance, no net movement of water in or out. the cell is happy. If you have way more stuff dissolved outside than inside, what do we call that?
Hypertonic.
That's going to be hypertonic. And what is that? What's going to happen to the cell?
Shrivel.
Shrink.
Yeah, the cell actually starts shriveling. Like we call crenation is when the little cells shrivel up because the water's leaving. They deflate basically. So these are three possibilities. In your hypotheses, you can say it's isotonic, the cells are happy. It's hypotonic, in which case they're swelling, and if they swell enough, they're going to explode. They don't have to, right? You could take a balloon and overblow it to where it's like really blown up, but it hasn't popped yet. But you blow it up even more and it pops. So just because it's hypotonic doesn't mean necessarily it's exploded, but it does mean that it's taken on water. So are there questions about these three situations here?
Can you explain why you use the term tonic versus osmotic again?
Yeah, so this, it's because of this thing I'm going to show you right now. So all of these cases, I'm talking about a non-penetrating solute. And in case of non-penetrating solute, anything hypoosmotic is hypotonic, isoosmotic is isotonic, hyperosmotic is hypertonic. But what if it's a penetrating solute? And so here we're going to see something really kind of weird happening. So what kind of things penetrate?
Non-polar stuff like fats.
So fats, alcohols, we're going to have glycerol is a non-polar thing, or has the ability to get into non-polar things, and also really small polar things. When we look at urea, urea is going to be penetrating because it's polar, but it's very small. So I'm going to put my, I'll be clever here and I'll use yellow here for 0.3 osmolar urea. So I have my cell.
It's like we don't have.
What would I call this in terms of osmolarity?
Isoosmotic.
It's isoosmotic solution. So I am putting my blood cell into an isoosmotic solution, but all is not well for my poor little cell. That rhymed again. In a healthy cell, urea is a waste product. Are we going to find urea inside a healthy blood cell?
No.
No. So we have a lot of urea floating around outside. There's no urea in the cell. Which way does the urea want to go?
Inside the cell.
It wants to move in, and it's a penetrating solute, which means it wants to, and it does. So all of a sudden we start having urea starting to enter into the cell. What is now happening to the osmolarity inside my cell as we start adding more solute to the stuff that's already there?
Increases.
Yeah, so now we're going to start increasing the osmolarity inside. And then what's going to happen to the water now as the osmolarity starts going up in here?
Goes inside.
The water's going to start going inside. So this is actually hypotonic, even though it's isoosmotic. So that's a little, so that's where you get that disconnect. It only happens when you're looking at these penetrating solutes, but that's why we have to have them separate. One is which way tonicity has to do with, does the water move or not? The osmotic is just, how do the numbers compare inside and out? Yeah, so I'm going to leave you, I realize I've gone over a little. I'm going to leave you with 4 your hypotheses. So 1 for, whoops, there's going to be 9 tubes that we're using, 1 for each solution, 9 in all. In each case, what are the 2 things you need to know in order to come up with a hypothesis about what's going to happen to the blood?
Dependent in
What is the concentration of each solution?
So, and I'm gonna give you the concentration. So what do you need to do beyond just pure concentration?
The osmolarity.
Yeah, you're gonna need to calculate the osmolarity. You know, and compare. Compare to what?
The red blood cell.
To the red blood cell.
Which is
0.3.
0.3.
Right. You're going to have to figure out what is the osmolarity of the solution and figure out, is it hypoosmotic or isoosmotic or hyperosmotic? What is the one other thing we need to pay attention to? The other independent variable.
The type of solute?
Yeah.
If the solute is polar or not polar type of solute.
And even more generally than that, whether it is what's affecting this whole system here.
If it penetrates.
penetrating or not.
Yeah, whether it's penetrating or not. Right, because we have, if it's not penetrating, then it's easy. If it's, you compare inside and outside, figure out if it's hypo, iso, or hyperosmotic, and that's going to tell you what happens to the cell. It's going to shrivel if it's hyper. It's going to be happy and stay the same. If it's iso, it's going to have water. It's going to inflate and maybe explode if it's hypo. As long as it's non-penetrating. If it's a penetrating solute, then you've got this weird situation where it can be isoosmotic, but it can still be hypotonic and explode.
So do you want us to just like list each one out?
Yeah, for each test tube, with each solution, you need to go through. So 1, for each solution, you need to calculate what is the osmolarity for that tube compared to here. Is it going to be hypo, hyper, or iso? And then also know whether it's penetrating or not penetrating. And therefore, you can then predict what you're going to find. Does that make sense? So cool. And I will put something up. I think I also have a little video I made kind of going over this again, which I will also put up. And that way you can review this. And sometime between now and Tuesday morning, I would like you to go through each of those 9 test tubes for lab, the fluid balance and RBC lab. And in your lab book, come up with your prediction for each tube.
So your predictions could be like, you know, because something is hypertonic and has this osmolarity, blank will happen.
Correct.
Okay.
Yeah,
I
it's
do that
penetrating and it's hypotonic, and therefore, you know, it's going to likely explode. But you won't be able to know if it's hypertonic or isotonic unless you actually calculate the osmolarity of the solution. And again, for that, you need to worry about, is it an electrolyte or is it not? So, I realize I've gone over 15 minutes, so I don't want to hold you all here. Any questions on this? Oh, Lauren?
So the lab that we just wrapped up today with like the demonstration stuff, is that also due on Tuesday?
That'll be due. Let's let that be due a little later than that, because I haven't even finished grading the chemistry lab yet. So let's make that due like in a week, another week after that.
So okay, so the following Tuesday, not that
Yeah.
Thursday.
Yeah,
Cool.
and I'll put that out there as well.
Okay, and then the graphs for the other lab are just due for for Tuesday.
This coming Tuesday.
Okay.
That's the jumping lab?
The jumping,
Yeah.
just to make sure people are staying, and if you don't get something done in time, you still want to do it and turn it in. But I am putting these deadlines on there just to make sure people don't end up digging themselves in this deep hole that they're going to have trouble digging themselves out of. And so if you continue working on these things consistently, then you have them done, rather than have this bunch of work piling up that you still have to get to.
So Professor, just go over one more time. Jumping lab, all the data due Tuesday, and then the ones that we went over today, the different diffusion ones, when are those due?
That'll be like in a couple of weeks.
And then this one also in a couple of weeks?
This one, all you need to do is come up with your pre-lab for Tuesday. You need to have a pre-lab for the new lab. So you need to have a hypothesis. Again, this will take you a little longer than your last hypothesis because there's 9 of them. And you have to do a little math, right? In order to calculate osmolarity, you actually have to do a little math compared to the molarity and know whether it's going to dissociate or not. But the math has to do with like dividing by 2, actually multiplying by 2, because we're going from molarity to osmolarity. So the math you can do in your head. And I will, after I'm done with the office hour stuff, I will go in and I will try to clean up the things to turn things in and make sure all the dates are clear. Maybe even like send out an announcement that has things more clearly stated so it doesn't feel as scattered.
That would be great.
Yeah, thank you.
Sure.